Good morning, it's Dr. Ray. I'm here to talk to you about the short version of development of the ear so that we can do interactive class activities during our in-class session. So ear development begins at about 4 weeks, and the area that the development is going to begin is the area of the developing hindbrain. Here you can see a cross-section of the embryo. And so this is about where the section is going to be, and you can see the developing neural tube, you can see ectoderm, and you can see a layer of mesenchyme that is going to be derived from both mesoderm and also neural crest cells. The surface ectoderm is going to thicken in that area and is going to develop into what we would call an otic placode. So here you can see that it's going to be developing right at about the area where the hindbrain will develop, and we'll pick this up when we do brain development, but that's going to be around the hindbrain, which we also refer to as the rhomencephalon. And so the optic cup is also going to develop from the brain region, and so both the ear and the eye, which have special senses, are going to be developing in this from this area. It's just that the optic placode is going to develop in the area where the diencephalon is located at, so it's going to be much more rostral, if you can see, and then the otic placode is more caudal in the developing neural tube. And so the otic placode is going to invaginate and create an otic pit. You can see here it's still made up of surface ectoderm, but it's going to invaginate. This is what it would look like in a scanning EM here of this embryo. You can see the location and the invagination of the surface ectoderm. So the otic pit, the two ends come in close approximation with each other and fuse and create an otic vesicle. That seems to sink down inside the mesenchyme. Really, it's just from differential growth of the embryo. But you can see how this otic vesicle gets surrounded by mesenchyme, and the surface ectoderm gets reunited with the two ends and becomes a continuous layer again. So this is what it looks like when you look at a histology picture of this otic vesicle kind of sinking down inside the mesenchyme of this region. There is a little bit of continuation from the otic vesicle towards the surface ectoderm, and that will develop into the endolymphatic duct. Now, this otic vesicle is going to become the membranous labyrinth of the inner ear. So when you learn the histology of the ear, and you were learning about the sensory receptors that are in the cochlea and the sensory receptors in the Utricle and Saccule for vestibular functions, you were learning about the tissues that came from this otic vesicle, that came from this surface ectoderm. So the otic vesicle is going to increase in size vertically and will develop into two subregions. The dorsal portion we refer to as the utricular portion, and that portion is going to develop into the utricle. And the ventral portion is the saccular portion, and that will develop into the saccule. So I'm going to talk about the two portions a little bit separately. The dorsal portion, which is going to develop into the utricular portion, you can see is going to develop into the semicircular canals and also the utricle. So the utricle itself is connected to the semicircular duct inside the semicircular canals. How those canals or ducts are formed is that first it's an outpouching, but then the middle portions of the outpouching, the ends come into close approximately with each other and fuse, and they undergo apoptosis, and so you end up with just a duct instead of a big flat disc. So it's kind of cool how they develop out of one big flat disc into three semicircular ducts. The ampulla of the semicircular ducts are just a dilated portion of the semicircular duct where those sensory receptors are located that you learned about in in histology where the crista ampullaris are located at. So the utricle and the saccule are continuous with each other. They come a little bit subdivided from each other though, and the saccule is going to be the part of this. Oops, sorry. I'm trying to think of the word. The part of this tissue that came from the surface ectoderm that is going to be connected to the cochlea. And so you can see the utricle that used to be all approximately this size, and then expands and grows out at a vertical dimension, and the saccule is going to be connected to the cochlea. So here you can see how this cochlea expands and projects outward and also curls in on itself about 2 3/4 of a turn to make the membranous portion of the cochlea. All right, so just to recap, at first, the otic placode forms, which is a thickening of surface ectoderm. The placode then invaginates and forms a pit. The otic pit becomes a vesicle as the ends fuse and it sinks down inside the mesenchyme. And then this otic vesicle will expand and grow into 2 sections. The dorsal section is going to become the utricle, and the ventral portion is going to become the saccule. And those portions are going to elongate and create the entire membranous labyrinth, which has the ducts inside that house the sensory receptors. So they actually have these sensory receptors in the walls of this tissue. And so you can see the semicircular ducts here, and you can see the cochlear duct here, and the saccule and the utricle, and the little endolymphatic duct. Okay, so we just talked about that membranous labyrinth, that's in blue. The bone that forms around the membranous labyrinth is going to undergo endochondral bone formation. So at first there will become a cartilaginous capsule that will form, and then the cartilaginous capsule will be replaced by bone. So in this mesenchyme, you'll also have little vacuoles, spaces that develop in the mesenchyme, and then they coalesce with each other, and they create a space around the membranous labyrinth that will become canals. So when you talk about the bony canal and these spaces, this paralymphatic space that develops, that's made from bone and then the space within bone, and that's in contrast to the duct, the membranous portion of the cochlear duct, where you see the cells in histology. And so here you can see how this happens in the cochlea, and you can see these the scala vestibuli developing here, the scala tympani. Here you can see the actual organ accordi, where that is going to develop at. And so here you can see the organ accordi developing. This is the tectorial membrane. Here you can see scala vestibuli. So you have this duct that's suspended in this bony labyrinth. Okay, so the bony labyrinth is a result of the otic capsule undergoing endochondral bone formation. The membranous labyrinth is going to be from surface ectoderm, all in blue. The outer bony labyrinth is derived from mesenchyme, which comes from mesoderm and neural crest origin, that undergoes endochondral bone formation. Okay, remember those pharyngeal arches that we talked about earlier, a couple classes ago? Remember that you have a surface ectoderm, you have a mesenchyme that comes from mesoderm and neural crest cells, and then you have endoderm that's on the inner surface of these pharyngeal arches. And that on the outside, you have a few places where they comes in close approximation, the ectoderm comes in close approximation with the endoderm. You still have some mesenchyme in the middle. And we call this together a pharyngeal membrane. On the outside, you can see how it grooves down, so it also could be called a groove. The arch is the big part that pouches out, and the inside where it comes inward towards the outer surface is called the pharyngeal pouch. So that's how, just as a little recap, we talk about the different structures. Well, how this comes into play with the ear is that the formation of the tympanic cavity itself, the middle ear, comes from the first pharyngeal pouch. So you see the pouch. Here's the first groove, and the pouch comes upwards towards the groove. So altogether, this would be a membrane. and it expands outwards and all the way to the groove, and you can see how the tympanic membrane is going to develop in this location. The pouch is going to expand and create the entire middle ear cavity, and it's still going to drain through the auditory tube or eustachian tube into the pharynx. So this is a really cool part of anatomy that is supposed to develop just this particular way. You can see where the inner ear that came from surface ectoderm that sunk down into the mesenchyme, it actually is in this location, and then the first pharyngeal pouch comes up and actually it ends up in between where that otic vesicle developed. And then here you have the first pharyngeal groove that makes the external acoustic meatus, and you have the tympanic membrane that lies between them. So it's a really cool developmental anatomical place. And so this is the specific part that we call the tympanic cavity, which is also called the middle ear. And the pharyngeotympanic tube will become more constricted down than the rest of the tympanic cavity there. And then after birth, you'll actually have some more mastoid air cells that will develop as we have a little bit of invasion of air cells in the mastoid process, because the mastoid process hasn't even finished developing yet. And so what happens is that this epithelial tissue that was from the pharynx, the cuboidal tissue, is going to actually not only come into the middle ear and cover the surface of the tympanic cavity, but also covers the surface of the ear ossicles. And then just as a little recap, that the malleus and incus come from the first pharyngeal arch, and then the stapes comes from the second, the first one being 5 and the second one being 7. Also the sapedius muscle might help you remember that that goes along with the 7th nerve, cranial nerve facial. Okay, and some other, just as a recap, some other pharyngeal arch derivatives in the middle ear. I like this picture because it kind of puts everything together. Here you can see the malleus and incus from arch 1. The mandible doesn't exactly, the actual mandible, there's a precursor, cartilaginous precursor. The mandible itself doesn't come from the arch cartilage, but in all intents and purposes, you can kind of think of it that way. The stapes, styloid process, stylohyoid ligament, top of the hyoid bone, this is all 2, or the facial. And then 3, the hyoid bone, the inferior aspect of the hyoid bone here, so going along with the glossopharyngeal. So you have V3, cranial nerve 7, and your cranial nerve 9. Okay, and so I just sort of laid it out here as like a little study guide, and highlighting here that first tympanic membrane is going to become, the first pharyngeal membrane is going to become the tympanic membrane, and you get a little bit of, you have thin skin on the outside because it's surface ectoderm. You have a little bit of connective tissue in the middle when you look at a histology picture of the tympanic membrane. And on the inner surface, you usually have a little bit of cuboidal cells that are coming from, derived from the first pharyngeal pouch. The external acoustic meatus is really an extension of the first pharyngeal groove, so it's going to have thin skin. The auricle itself actually is going to develop from these auricular hillocks that develop, and they are from the first and second pharyngeal arches. So you can see they number them, and 1 and 2. They don't exactly line up exactly perfectly when you look at the finished development of the ear. They don't sort of line up in a great easy lines, but you can see how you could have a lot of malformation of the auricle due to lack of proliferation or lack of fusing of these auricular hillocks. So there's a lot of variation in the way that ears are formed and shaped. And so here you can see what it looks like in an actual embryo developing, pharyngeal arch 1 and pharyngeal arch 2, in the orientation of where those auricular hillocks are located. Okay, so that's it. I hope you understand what you need to understand about development of the ear for our session, and if you have any questions, feel free to contact me.